All right, today we're putting on our detective hats. We're going to be on the trail of a biological mystery, one that stumps scientists for more than 100 years. And we're going to see how cracking just one single clue completely changed the game for a really devastating disease. Okay, let's dive in.
So just imagine for a second, what if the messengers that carry instructions from your brain to your muscles, what if they just started breaking down, going rogue, delivering toxic packages instead of the right orders? Well, that's the core problem we're investigating today. And that brings us to our case file. The central mystery is a disease that for decades had no clear cause and, honestly, no effective treatment. A total enigma. Now, the official name is amyotrophic lateral sclerosis. You've probably heard it called ALS. The easiest way to think about it is this. Your motor neurons are like a huge fleet of delivery trucks, constantly carrying orders to your muscles. In ALS, these trucks just start breaking down, one by one, until the messages, they stop getting through at all. And the consequences are just devastating. As those messengers die off, people slowly lose control over their own bodies. It impacts everything, moving, speaking, even breathing. And for the longest time, the reason why this was happening was a complete blank spot on the map. I mean, the stakes for solving this case could not have been higher. But then, after a century of searching, investigators finally got their first major break, a single clue that would blow this whole case wide open. Just look at this timeline. It really shows you how long this mystery went unsolved. It was first described way back in 1869. It took over 100 years to find the first real lead. Then, finally, in 1993, by studying the DNA of families where ALS was common, scientists pinpointed their prime suspect, a single gene called SOD1. And this SOD1 gene, it was the breakthrough. It was the first hard proof that ALS could actually be genetic. Now, it only explained a fraction of cases that run in families, but that didn't matter. It was the thread that investigators could finally start to pull on to unravel the entire mystery. So once you've got a suspect, what's the next step for any good detective? You've got to figure out the motive and the method. How exactly does this one gene cause so much destruction? Well, in its normal, healthy state, the protein made by this SOD1 gene is actually a hero. A superhero, really. Its job is to act like a cellular firefighter, putting out these dangerous chemical sparks called free radicals before they can cause damage. It's absolutely essential for keeping the cell clean and safe. But a mutation changes everything. The hero turns into a villain. And here's the crazy part. Instead of just failing at its old job, the mutated SOD1 protein gains all these new toxic abilities. The protein, which is supposed to fold like perfect origami, now just misfolds into a crumpled, unstable mess. And these messes, they start sticking together. I saw one description that just nails it perfectly. It says it's like pouring glue into a bowl of spaghetti. The strands stick and tangle. And that's exactly what's happening inside the neuron. These misfolded proteins form these sticky clumps, these aggregates, creating a disaster that just gums up the works of the entire cell. So here's the path to destruction, step by step. It all starts with a typo in the genetic recipe. That leads to a misfolded protein. Then, these proteins form those sticky clumps. Those clumps then attack the cell from the inside out. They disrupt its power plants, they basically poison it. And ultimately, the whole system fails and the neuron dies. This whole process is what scientists call a toxic gain of function. But cracking this one case did so much more than just explain the method. Discovering SOD's role created this huge ripple effect that would change the entire field of neurological research forever. I mean, finding SOD1 was a revolution. It proved ALS wasn't just some random bad luck. It could be genetic. It identified protein misfolding as a key mechanism, which suddenly linked ALS to other diseases like Alzheimer's and Parkinson's. And crucially, it allowed scientists to finally create animal models to actually test therapies. It just kicked off this genetic hunt that has since uncovered over 40 other genes linked to ALS. And that brings us to the final chapter, the best part. With the culprit identified and its methods totally understood, scientists could finally move from just investigating to intervening and bring about a whole new era of hope. The new strategy was brilliant in its simplicity. I mean, why just clean up the mess when you can stop the villain from making it in the first place, right? The goal became to target the root cause by silencing the faulty SOD1 gene before it could even produce the toxic protein.
And the results of this strategy? They have been absolutely stunning. A therapy called tofersen, which uses a special tool to silence that gene, has been shown to reduce the levels of that toxic SOD1 protein by up to 70%. That's a massive reduction.
And just look how quickly this has moved from the lab to actual patients. After it got approved in the US in 2023, later data showed it can actually slow the disease down and even help people regain function. This isn't just about managing symptoms anymore. This is about changing the entire course of the disease. And you don't have to take my word for it. You can literally see the proof in the biological data. This chart shows the levels of a key marker for nerve damage. It's called NFL. And in patients treated with tofersen, well, those damage markers just plummet. It's clear, visual proof that attacking the source really works.
So the detective story of SOD1 really shows us that a single clue can unlock decades of mystery and lead to real, tangible hope. The blueprint that was developed here, from finding the gene to creating a targeted therapy, that's now being applied to other genes and other diseases. The code has been cracked for this one culprit. So the only question left is, which one is next?